So let's move to the last unit I wanted to talk about here, which is now that we've got a big pile of well-calibrated flow cytometry data, what do we want to do to visualize and interpret it? So let's start with a reminder that we're going to be thinking about things not in terms of ordinary means and standard deviations, but in terms of geometric statistics, which are essentially means and standard deviations on the log scale. You may have noticed that almost every graph that I've shown so far is using log scale axes. And that's because our cell expression tends to be bell curves on the log scale. And there's a reason for this. This comes down to essentially that anything to do with gene expression can be thought about as a complex reaction with a whole bunch of rates being multiplied for all the stuff that we usually don't model, like the actual transcription and translation processes, which when you multiply a bunch of rates together that all might vary a little bit, that should lead you to expect things that look like nice normal bell curves on the log scale. So we're going to work with geometric statistics in analysis. A reminder about how those get computed. They're really just your normal arithmetic statistics, means and standard deviations on a log scale. The key consequence of this is that your error bars are times and divide rather than plus and minus. And on the bottom right here is an example of showing how a geometric statistics fits very well with this flow cytometry data from a TED-R device, and an arithmetic fits very, very badly. So there is some example data that we've put up that you can play with and analyze. So if you go to the iGEM measurement tools on GitHub and look in fluorescence tutorials, there is a collection of flow cytometry data with a set of things applying the TASB flow analytics package. So the color model directory is setting up the calibration, and the analysis directory is using that calibration in order to interpret a bunch of data that's in this data set from 2014. This particular project, this is one that was being done by Swati Kaur, then of the Densmore Lab, with one of these very sort of standard inverter circuits. This was an Arabinose induction of TED-R circuit where we were studying the input-output relationship of TED-R. I think this is the sort of thing that I suspect is a simple relationship that you may see in a lot of iGEM projects. Some example of the data coming out of here. So we're essentially, the Arabinose induction is showing up in green. So we should expect to see low green when there's low Arabinose and high green when there's high Arabinose. And that Arabinose is inducing also TED-R, which then represses red. So we ought to see that when there's low Arabinose, there's low TED-R and therefore high red. And when there's high Arabinose, there should be high TED-R, which then turns off the red, and the red should be low. And here we can see that the green on the left is much lower than the green on the right, although there's a bit of a smeariness off to the left here, so there's some cells that are never really turning on. And the red on the left is a nice sharp peak, not very high, but on the right, where we should be turning off the red, most of the red turns off pretty well, but there are some cells that really aren't quite turning off. So let's talk about how we interpret information like this. We can take this, and look at the whole range of information. We induce this at a whole bunch of different levels. So we can take these input-output information, plot them up as calibration curves. So here we can see on the left, at different dosages of arabinose, how much GFP is coming out. And the error bars I plotted here, they are not the per replicate differences. These replicas are actually very tight. As going back here for a moment, you can see there's multiple replicates being plotted. These are graphs coming directly out of TASB. There's 3 replicates on each graph, and they're very, very similar. What we're plotting here is the error bars instead is the cell-to-cell variation within samples. And the reason that's important to know is because if we want to talk about something being effectively separated in its on state from its off state, we want to know how well distinguished are the cells. Because the differences happen within a cell, not across the means when we're hooking up parts within a cell. So we can see here that for the arabinose at least, this, these off areas are actually pretty different from when it's full on. Even the low cells are pretty far away from the high cells in the off state, although the middle is pretty broad. So we've got to get all the way out to the ends to really get nicely distinguished arabinose. For the TET-R, on the other hand, the input versus the output are full on TET-R. Still, these cells are overlapping a good deal with the high cells from the full off. So let's think a little bit more about how to be systematic about understanding what's going on here. And what does this tell us for models and for devices? One of the nice tools that we can bring in is an idea of signal to noise ratio. This is an information tool that has been long developed in the electronics and communication and that whole world. And it simply asks, how well can you distinguish 2 signals? Those signals don't need to be electronic or radio signals. They can be a signal like the fluorescence signal outside of coming out of our cells. So if we say, here's the distribution of offs, and here's the distribution of ons, signal to noise ratio is going to help us understand if I pick a random cell out of the off, how easy or hard is it going to be for me to tell if that's really out of the off versus out of the on? So if I get a random cell from over here, it'll be really easy for me to tell that that's not out of the on distribution. But if I get a random cell over here, well, there's some overlap, and it may be a little bit difficult to tell. So we can estimate these numbers in decibels just by using the geometric mean and standard deviation statistics if we've got some reasonably bell-shaped curves. It gets a little bit more complicated when you have things that are bimodal. So we can ask, how clearly can we distinguish things? And if we look at how clearly can we distinguish outputs versus how clearly can we distinguish inputs, that can give us a change in SNR, which tells us things about the type of circuits we can potentially build with the device. So applying this to our device here, we can see that the P-bad induction has the high and low are very clearly separated. When we apply a signal to noise ratio calculation to it, we can see we've got nearly 6 decibels, that's a pretty good number. That's a number that you can engineer with. This decibels, the same sort of decibels that are telling you like how loud and how clear is the music you're listening to. So that's telling us this is working pretty well. Over in TET-R, we actually get a surprisingly precise 0 decibels comparing the high and the low. That's telling us that the difference between those peaks is the same size as how sloppily wide the peaks are, which means that this is, we can distinguish it, but it's going to be hard to do much engineering with them. So the sort of some conclusion we can draw from this is in this particular circuit, our arabinose induction worked pretty well. We can work with that. TET-R, not working so well. And if we look at, because we've calibrated this into absolute units, to these equivalent fluorescein units, we can also start making some guesses about what might be going wrong. Because these units, molecules of equivalent fluorescein, is pretty close to molecules of GFP. And since we're working in E. coli, we know how many molecules are reasonable to try and get. We can try and get 10 to the 5th molecules pretty easily within an E. coli, which has about 2 times 10 to the 6 molecules all told of protein. That's a number that comes from bionumbers.org and that we talked about some in the seminar last week. So we can see that this p-bad is going most of the range, although it seems to be a bit leaky at the bottom, that 10 to the 3. We might be able to get lower than that. TED-R, on the other hand, is never really turning on all the way. And that gives us a clue as to what might be the problem. It's nice and tightly off when the TED-R is high. That 10 to the 2, that's pretty close down near the autofluorescence limit. This black bar here, that's coming out of the autofluorescence model from the calibration. So this is pretty low. We're never really turning it on all the way, and that gives us a clue as to what our models might want to be applied to try and figure out how to re-engineer this device to get a better TED-R. So, in summary, if we use geometric statistics in summarizing flow cytometry data, that can give us a good idea of what's going on across those big, broad distributions. We, of course, if we're looking at a complicated distribution and trying to make a complicated model, we may need to go deeper. But for a lot of simple engineering things with bacteria or yeast or other simple cells that are often used in iGEM, simple geometric statistics will get you a long, long way. You may want to dig deeper into the particular distributions of your single cell behaviors. And if you're looking at devices that you're trying to switch on and off, signal to noise ratio is a very useful tool for assessing them. And the fact that we've got calibrated measurements that we can relate to molecules can help us interpret what's going on in our measurements with respect to biological intuitions and help suggest approaches for improvement. And with that, we've come to the end of what I wanted to share with you today. I'll be happy to take any more questions before we wrap up. All right, seeing no questions, we're going to wrap up here. I want to remind people that the measurement committee is also doing office hours. There is an office hours at 1 p.m. Boston time today. And I see a question. So sometimes we see 561 something A on a y-axis and 488 something A on an. So that 561 and 488, those are nanometer numbers for the lasers. So the actual names you get for the channels are completely arbitrary. The names are just set by whoever set up the instrument. People often like to put either the name of the fluorescent dye that they're calibrating towards, like fluorescein or FITC, Texas red. They'll often like to put the laser emission number. So like 561 is a laser that's typically used for measuring red fluorescence. 488, that's a blue laser that's typically used for measuring yellow or green fluorescence. And then the dash A, that would mean area. So dash A, dash H, dash W. C. And second question from Hassan is, if I understood you, mostly were you interested in the area, not the height or the width? That is correct. In fact, it's not just iGEM projects. Most projects, usually if we're interested in fluorescence, we're usually interested in the area and not the height or the width. And the height and the width are used mostly for gating. I have seen exceptions. So like, for example, when somebody was studying a cell killing process, they really wanted to look at the other things too, because they wanted to actually study the debris itself directly. So hopefully that's helpful. So I'm just going to wrap up by noting that, again, that we have an office hours that people can come to. You can also submit questions to us via email or via, there's a survey link on the Measurement Hub on the website. All of this stuff is going up onto the measurement webinars page, both the PDFs of these talks and recordings when HQ finishes processing them. If you go to GitHub, to the iGEM measurement organization, you will find all the example fluorescence data from both this talk and the talk last week, and MATLAB/Octave code for working with it. Thank you, everybody, and I hope that this has been useful.